Okay, well, everyone, we might as well get started. It's right up on 2 o'clock. So thank you all for joining us this afternoon for our 22nd session of the Journey Down the Clackamas. And I'm Dave Bugney, a board member of the Clackamas River Basin Council. And joining me this afternoon is Gail Shalom, who is the CRBC board chair, just like she said, for the next 3 days, who will assist me in the technical aspects of our program this afternoon. So before we get into it, I will cover a few items. So the Clackamas River Basin Council is celebrating its 25th anniversary. Since its founding in 1996, the CRBC has worked as a non-governmental group of stakeholders to protect and improve the Clackamas River Basin and its tributaries. Their mission is to foster partnerships for clean water and to improve fish and wildlife habitat and the quality of life for all those who live, work, and recreate in the Clackamas River watershed. So on behalf of the Basin Council, I'd like to thank our two gold sponsors, the Oregon Beverage Recycling Cooperative and the Clackamas Water Environment Services. And we'd also like to thank, there's probably going to be a little video that's going to come up, but it's going to stop right there. Good job. And we also thank our bronze sponsors, the Clackamas River Water Providers, the Geological Society of the Oregon Country, Port Blakely, and Metro. So we'd also like to thank the many individuals who have donated to support this conference. And we continue to seek additional donations, which are tax deductible. You got, several more days to really make that big donation. So for this year, anyway, the Clackamas River Basin has been occupied by Native peoples for millennia. It's originally the territory of the Clackamas, Chinook, Molalla, Kalapuya, and other peoples, and is currently recognized as lands of the Confederated Tribes of the Grand Ronde, the Confederated Tribes of the Warm Springs, and belongs to other Native peoples, may not be federally recognized. And we thank those who came before us for their stewardship of these lands and waters, and those who continue to steward them now and in the future. So we're gathered for this journey down the Clackamas to learn in-depth information about the basin and its connections to Oregon as a whole, to connect with each other so that we can support our mutual efforts to protect and improve the Clackamas River and its tributaries, and to have some good old-fashioned educational fun, even during winter break. So CRBC is partnering with the Environmental Learning Center at Clackamas Community College. So workshop attendees will receive a signed certificate of completion for each session. If you'd like a certificate of completion, please add your contact information to our Journey Down the Clackamas Conferencing Networking Google Sheet and indicate you'd like a certificate in the G column, and we'll forward your contact information on to Clackamas Community College. So I'd like to share a few ground rules before we introduce our special guests. So please keep your microphones on mute unless invited to participate. And you can keep your cameras off if you like. That'll help us save bandwidth for our presenters and greatly reduce the carbon footprint of our meeting. We'll have two presenters this afternoon with a brief Q&A after each presentation. And so please type your questions in chat and we'll select the questions from there. But if you have a real complex question or one that is rather long, feel free to just chime it in on audio at the appropriate time, and we'll get to it. However, if your question is, as I mentioned, please feel free to do it on audio. Feel free to send direct chats to friends you're seeing in the participants list. So we'll start our presentation now for this evening. So please remember to type your questions in chat, keep yourself muted, and we'll answer questions at the end of each presentation. The webinar will extend to about 3 p.m. this afternoon with questions afterwards. So our presenters this afternoon, first is, Ian Corder, who's a founding scientist with Mount Hood Environmental. He served as principal investigator for naturalist fish studies in numerous watersheds throughout the Pacific Northwest, including the Clackamas. He holds a BS in environmental biology from Pacific University and an MS in fisheries science from OSU. Our second presenter to afternoon is Maureen Kavanagh, and she's been working for Bonneville Power Administration since 2015. And she is the hatchery policy lead in Bonneville's Fish and Wildlife Program. Previously, Maureen worked for 11 years as a fish biologist for the U.S. Fish and Wildlife Service, Columbia River Fish and Wildlife Conservation Office, where she developed and implemented fish management plans to support hatchery reform and hatchery review team recommendations for best management practices at federal fish hatcheries. Maureen holds a master's of biology from Western Carolina University and a BA in environmental science from Warren Wilson College in Asheville, North Carolina. So now please let's welcome Ian and Maureen as they present about the interactions and behaviors of wild and hatchery-reared fish within the Clackamas River Basin. So take it away, you two.
All right, thank you. Yeah, my name is Ian Corder. I'm going to start off this afternoon session here, and then Maureen's going to follow. Let me get my presentation rolling here. Just give me 1 second. All righty, well, hopefully you all can see my title slide there well. Somebody let me know if you can't for any reason. Also, I'm assuming that David or someone will let me know if you have any difficulty hearing me. I'm getting a thumbs up, so we're good to go. So that's right, my name is Ian Corder. I'm a fisheries scientist with a small company called Mount Hood Environmental that my wife and I started about 10 years ago. And we specialize in original research projects, fisheries and water quality related projects. We also do a variety of service projects, invasive species removal, fish salvage efforts, and water quality monitoring work throughout the Northwest. And I have done a little bit of work in the Clackamas Basin specific to hatchery fish effects on natural origin winter steelhead productivity. And I presume that is why I was asked to give this presentation. This is going to be, as I would say, non-technical as I can make it. This is going to be a very high level, simple overview of my interpretation of the state of the science concerning hatchery fish, with a couple of specifics related to some of the work that I participated in the Clackamas Basin, but really would like to leave you with kind of a sense of, at least from my perspective, what the available peer-reviewed science on this topic would tell us about the types of issues to be aware of with respect to hatchery fish impacts on natural origin fish production. I'm gonna go to my next slide here, if I can figure out how to operate that. I think it's, besides being a scientist, I am a pretty avid angler. I've got a pile of kids at home, and we spend a good deal of our time trying to catch salmon and steelhead. The reason I think that's relevant to this presentation is really just my interest in this topic. This hatchery fish science topic is important to me because, on the one hand, if hatchery fish are impairing the productivity of natural origin salmon and steelhead populations, that would be a significant concern from the standpoint of just continuing to be able to participate in these fisheries. Sustainability of these populations is obviously crucial. So if hatchery fish are negatively impacting wild fish, we need to know that. On the other side of that coin, if there are scenarios where hatchery fish don't have a large impact on natural origin fish production, that is also very important because, as most of you I suspect know, there are a lot of hatchery fish out there supporting both not just sport fisheries, but also tribal fisheries and commercial fishing as well. So we're very dependent on hatchery fish within our fisheries. So whether hatchery fish are impacting wild fish or not is a pretty crucial question from a fisheries science standpoint, in my opinion. So first off, really basic, what is a hatchery fish? Well, it's a salmon or steelhead that has been spent part of its life in a hatchery facility in a raceway, as is pictured here on the left. That's an an aerial photo there of Eagle Creek hatchery. But so a fish that spends part of its life in a hatchery, typically a couple of months up to about a year in most programs, those fish are then released into a natural environment where they can complete their life cycle. And some number of them will then return to the hatchery to propagate the next generation. So that's what a hatchery fish is. Why do we produce hatchery fish? Well, I think it's fairly obvious. We modify our watersheds. This is just a picture of the Portland, kind of skyline there of Portland. And we make some pretty substantial modifications to the habitat that salmon and steelhead rely on. We build dams for power production and flood control and irrigation water diversion. And we also, it turns out, like to catch these fish, as I mentioned earlier. That's just a picture from 1920 on the Columbia River, some commercial fishermen harvesting salmon. And so with these kind of conflicting resource uses and objectives, hatcheries have been used for a long time to mitigate for the impacts of habitat modification and still accommodate harvest in commercial and recreational fishing.
Put on your jacket and help me put it back together tighter.
Not sure if that was for me or somebody else. Somebody's unmuted there. Just heads up. So the Clackamas River Basin is obviously has a long history with hatcheries. First hatchery was salmon hatchery in the Northwest was built on the Clackamas River back in the late 1800s. And of course, most of you know that today we have two hatchery facilities, one on Eagle Creek and then the Clackamas Fish Hatchery that produce salmon and steelhead in the basin. So concerning hatchery fish effects, so established thus far in the presentation that hatcheries have been used for a long time for mitigation purposes, and while they have been a pretty critical tool for management, they've also been implicated in the decline of salmon and steelhead populations, and some researchers have asserted genetic effects or discovered potential genetic effects. Others have documented potential ecological impacts. And then sort of less common in the literature, more so in kind of management documents, there are also potentially direct impacts of facilities on things like water quality and fish passage. On the genetic side of things, specifically, things like a domestication of hatchery origin fish has been documented as a key concern. So that issue is wrapped around the idea that as you bring hatchery fish into a hatchery, then generation after generation, those fish, like any animal, begin to adapt to a captive rearing environment, and genetically potentially will then favor traits that make them more effective at surviving within a captive rearing environment instead of surviving in the wild. And as those fish, to the extent that those are genetic traits that they've developed within the hatchery, as those fish then enter a natural environment, and some of them will stray and spawn with natural origin fish. And if those genetic traits get passed then into the natural origin fish population, that could cause a negative impact on wild fish. And I would say probably that some of the most widely cited studies or examples of that process occurred in the Hood River, Oregon. So Oregon State University developed a series of publications where they demonstrated how that domestication process could impact the fitness of hatchery origin fish. And they didn't necessarily show that that fitness loss was then inherited by the wild fish population, but they did demonstrate that hatchery fish were less reproductively successful in the wild. So that's a potential concern. On the ecological effects side of things, a specific example I would bring up there is some work that was done by Tom Nicholson on the Oregon coast, coastal coho salmon, where he demonstrated that there was a correlation between the abundance of juvenile hatchery coho salmon that were released and the survival of natural origin coho salmon. He found basically that the more hatchery salmon that were released, the lower the survival rate that was being observed in the wild fish population. And he asserted that there could be a mechanism whereby predators or something like that were being attracted to predate at a higher rate on natural origin fish when there was an abundance of hatchery origin fish in the population. So that's just one specific example of an ecological impact. So these are categorically, these are the kinds of ways that the folks have documented concerns about the use of hatchery fish with respect to recovery and enhancement of wild fish populations. So from my perspective, as I just look at a cross section of the various papers that have been published on the topic of hatchery fish science, what I find is an abundance of papers that document possible mechanisms for effects. And I just gave you a couple of specific examples of papers that have documented possible mechanisms for impact that we would regard those as evidence for potential risk of hatchery fish. There are theories that have been, I would say, fortified by the collection of empirical data, but not necessarily evidence of a direct impact. So they didn't necessarily quantify a direct impact on a wild fish population, but they inferred potential impacts through various genetic or ecological mechanisms. So there's an abundance of research that I would say falls into that category. And I think that that's why, in general, why hatchery fish are regarded as a risk to natural origin fish. There's also sort of another set of peer-reviewed studies out there that I would put into sort of the population level effects category. And there are fewer of these available to us. And we're seeing kind of a mix of findings. So in some cases, the qualitative conclusion of the paper was negative effect at a population scale on wild fish productivity or survival. In other cases, folks actually have demonstrated positive effects of hatchery fish, so demographic boost or a boost in abundance or productivity within the population as a result of a hatchery program. And then another set of studies that have concluded no quantifiable negative effect of hatchery fish. And these are the studies that I'm personally most interested in, because I think that taking all of those, all the previous sort of cross-section of literature that I showed in the previous slide highlights all the possible mechanisms. But what I'm interested in is demonstration at the population scale that hatchery fish have negatively affected wild fish survival or productivity. I think that these studies highlight the kind of the mix of outcomes here, negative, positive, and no effect, kind of highlights that there are certainly pieces of the puzzle sort of missing from the hatchery fish science conversation, things that are yet to be discovered, and how these hatchery and wild fish interactions really play out at a population scale. And I think this is a critical question for management, because this is ultimately what we want to know from a fish recovery standpoint, is how can we get to more robust fish populations, and to what extent do hatchery fish affect wild fish relative to other factors that are influencing abundance and productivity. So some of the reasons that I think there are relatively few case studies that fall into this population level effects category is that frankly, it's difficult. These studies require long-term data sets, and there's not a limitless supply of this kind of data to work with. And then there are also issues that I think sort of have come to light as I've reviewed previous studies that make it very difficult to tease out hatchery fish effects relative to other factors. And I think that's impaired our progress and ability to really hone in on the scope and scale of hatchery fish effects relative to other factors. So, for example, productivity estimates can be sensitive to the inclusion of hatchery origin spawners. So whether you choose as a researcher to include hatchery origin spawners along with your natural origin spawners when you estimate the productivity of a population, the inclusion or exclusion of those hatchery origin fish can have a large impact on kind of your inferences about the effect of those hatchery fish, because we know that as fish population dynamics biologists, we know that the more spawners on the spawning grounds, the lower the survival of their offspring due to density dependent survival effects. And so including or excluding those hatchery origin spawners can very much affect that calculation. We also know that, and I highlighted some of Mark Chilcote's work. Mark was a biologist with the Oregon Department of Fish and Wildlife, and then later with the National Marine Fisheries Service. And he published a couple of papers that I think highlight the importance of that choice to include or exclude hatchery origin spawners. fishing impacts and other, I would say more generally, impacts of factors that co-occur along with a major change in hatchery fish production. This is another sort of challenge that we face when we look at the available case studies for hatchery fish effects. And Eric Buell, who's a research affiliate with the National Marine Fisheries Service, published a nice paper on Oregon coastal coho salmon, where he did kind of a meta-analysis of coastal salmon on Oregon coastal coho salmon and the effects of hatchery fish. And he found that the abundance of hatchery fish did impact the productivity of Oregon coastal coho populations. The issue there, or the challenge there, though, was at the same time that hatchery fish, coho hatchery production had been eliminated, there were major changes in fishing practices as well as a whole lot of habitat restoration work that co-occurred with that change in hatchery management. And so that those co-occurring kind of management decisions, I think, confound the impacts, predicted impact of hatchery fish in that case, just simply because it's hard to pull apart those things when they essentially are happening at the same time. Then finally, another issue that I have noted in the literature is just the importance of reference populations. And there was some work done in the Clackamas, and I'm going to transition specifically to a case study in the Clackamas here now, using this third bullet point as kind of a launching point. But reference populations with respect to salmon and steelhead productivity analyses are very important, because we know that there are large sort of regional factors like ocean conditions or climate conditions, regional climate conditions, that have been associated with productivity across basins. And that there tends to be trends in abundance that across basins for both salmon and steelhead, that they tend to follow each other sort of up and down, and oscillate up and down together. And if that's not accounted for, if those regional impacts of climate and oceans aren't accounted for, then the result of that could be sort of a misunderstanding about what's driving the population up and down. Is it something like a change in hatchery management, or is it really something like a change in ocean conditions? And I want to highlight some findings that specifically from the Clackamas, where we learned that some of our inferences about the impacts of hatchery fish, the original inferences, weren't correct. And so I'll highlight this case study here, where we looked at winter steelhead in the upper Clackamas Basin and the impacts of hatchery origin summer steelhead. In this case, researchers from the Oregon Department of Fish and Wildlife found that, and I think that logically so, they looked at the abundance of natural origin winter steelhead at North Fork Dam. This is a pretty unique data set, because it started in 1958 and is still being collected today. So we have an opportunity here to, with a long time series, examine the effect of hatchery fish. In this case, a segregated summer steelhead program. And what the original researchers found was that there appears to be an impact of the beginning of this summer steelhead program in the upper Clackamas Basin coincided with a decline in winter steelhead abundance. And so that led to a productivity analysis and a conclusion, sort of a published conclusion in Transactions of American Fisheries Society, that winter steelhead productivity had in fact declined as a result of the presence of these hatchery origin summer steelhead. And as you can see, the number of adult summer steelhead returning to North Fork Dam was very high, as high and above 10,000 returning fish. That's a large number of fish relative to the size of that watershed. And so these were pretty compelling findings, and sort of widely cited as the basis for kind of a direct example of how hatchery fish can have a large negative effect on natural origin fish productivity. Garth Wyatt and I, there's Garth on the right, holding the Chinook salmon on the Deschutes River. But we decided to take another look at this data set. That original paper was published in 2006. And so we had about 15 more years of data to work with. And so we decided to kind of re-examine that data set along with other researchers from Portland General Electric and the Columbia River Intertribal Fish Commission, as well as USGS. And just in beginning that analysis, the first thing that we noted was that Clackamas winter steelhead abundance, again, these are counts at North Fork Dam, and the productivity of those fish, so recruits per spawner is the metric that I'm showing you here. If you look at the left-hand side, that's the number of adult fish returning per spawner that entered the upper basin. What we found was in our initial kind of reconnaissance effort was that Clackamas winter steelhead abundance, recruits per spawner, seemed to trend similar to recruits per spawner estimates from Willamette Falls. Shorter time series there, but similar to the Hood River, as well as the Sandy River. So the absolute values differ, yes, but the trends themselves kind of follow a similar pattern, and we thought that that was compelling. And this gets at the issue I mentioned earlier about utilizing reference populations when making inferences about what's driving salmon and steelhead population abundance and productivity. We also recognized that there was a great deal of harvest in the upper Clackamas Basin. So what this plot is showing you here is that same time series, the dark black line. The gray line is the number of adult summer steelhead returning at North Fork, and the dotted gray line is the number of summer steelhead that we estimated to be in the upper basin after accounting for harvest. So anglers actually harvested a large number of those summer steelhead, and we were able to obtain that data from Oregon Department of Fish and Wildlife, the catch card data. So if you adjust the number of summer steelhead that were up there available really to interact with wild fish, it's still a significant number, but far less than the number returning to North Fork Dam. Another thing I'll point out just from this plot, and it's sort of cut off on my screen just due to the tiles over here on the right for the participants, but I believe that what's being shown here as well is that black line extending on the time series out to about 2019. And as you can see, the trend in abundance doesn't necessarily pop right back up after that hatchery program ends. It continues to bump along around a stable equilibrium of somewhere in 1,000 to 1,200 spawners or so for that population. And so I think that was kind of another important anecdotal piece of evidence that we had to suggest that this data set needed further analysis. We also knew that there was not a complete overlap in where juvenile summer steelhead were being released relative to where natural origin winter steelhead were spawning in the upper Clackamas Basin. And so this figure here, which is from a paper that we published in Transaction of American Fisheries Society, highlights that the fact that summer steelhead releases generally were occurring lower in the basin at lower elevation relative to where the natural origin winter steelhead population was spawning. The reason that last little bit there was significant is that, and I'll just hit you with kind of the high level takeaways from that paper. But the reason that was important is that we asserted that there was separation in spawn timing and location between the summer steelhead and the natural origin winter steelhead in the basin. And we thought that might explain why we were unable to quantify a negative effect of hatchery origin fish on winter steelhead production in the upper Clackamas. We did, however, quantify other freshwater and ocean survival covariates that did appear to be driving winter steelhead abundance. And then again, we documented the synchronicity between steelhead populations in the region, again, just emphasizing that there are these sort of broad scale variables that seem to be driving abundance to a greater degree than the presence of hatchery fish. In the future going forward, and again, that's, I'll say just kind of as one side note to that last bit about the study that Garth and I led there. I would say that, you know, that's a very specific case study. That was a segregated summer steelhead hatchery program. There's an abundance of other issues, particularly in the lower basin, that likely still need to be addressed and studied. I would say that in general for hatchery fish science, what I'm most interested in is a continued effort to quantify these population level effects of hatchery fish relative to other factors. I think that those studies help us, will help us kind of emphasize the pieces of the puzzle that we think we might be missing in terms of our understanding of where do hatchery fish work relative to in terms of minimizing their impacts on wild fish, and where might they not work as well. Another area of research that I'm interested in is the differentiation between hatchery fish impacts and the impacts of hatchery management. I think that's been somewhat muddled in the available scientific literature, where it's hard to tell, especially for the lay public. It's difficult to discern whether a researcher is defining an impact of something that's under management control, like the way, for example, the way that broodstock is collected, whether you use in-basin or out-of-basin stock, and when and how you collect those fish. You know, that's something that's under management control, but often gets, I think, misrepresented as a direct impact of hatchery fish. And those things, that's sort of a nuanced issue, but they're distinct, and I think they need to be better defined in the literature. Then lastly, I'll just say, I'm hoping going forward that there will be greater collaboration between fisheries biologists, fisheries population dynamics, biologists in particular, and geneticists. For the past, 10 to 20 years, genetics has really dominated the hatchery science conversation. And I think that it would be really valuable to see a sort of a better blend of fish biology with fisheries genetics to arrive at a better understanding of what the impacts of hatchery fish are on natural origin fish production. So I believe that that's my last slide. It is. I'll stop sharing here. Thank you for your time this afternoon. I hope that that was somewhat informative, and I'll take any questions if I have time, David.
Thanks, Ian. Yeah, if anybody has any questions they want to put in chat, or if the question is too complicated to phrase in chat clearly, feel free to just speak up. Remember that Maureen will also be presenting further, so if you want to hold your questions till after Maureen is done, that's certainly fine too.
I don't see any questions right now in the chat.
Okay, no problem. We'll have time after Maureen is done to ask any questions that you might have, either about Ian's presentation or Maureen. So with that, why don't we go ahead and get Maureen going?
Hi, yeah, thanks. That was a great presentation, Ian. I think it's a good segue into talking about some of the specific hatchery wildfish interaction studies that I was involved in Eagle Creek National Fish Hatchery and in the Basin. I'm going to go ahead and share my screen. Let's see. Get this. Okay. Okay, great. Can you all see this okay?
I am not seeing your screen.
Okay, let's try this again.
We did have a question come up. Maybe while Maureen is putting up her screen, Ian, if you have a minute to answer this. Maddie asks, can you please define your use of the word recruits as compared to spawners?
Oh, sure, no problem. So recruits, that's a good question, and I probably just did a terrible job of explaining that. It's fairly important. So recruits can be defined really as any life stage within the life cycle, but it's a reference to the number of surviving offspring from a spawner. So in other words, if a female steelhead produces 2 fish that survive the entire life cycle and return as adults, those would be regarded as adult recruits from that individual steelhead. So when we use a metric like recruits per spawner, that's what we're getting at. And for the purposes of the analysis I was describing, those were adult recruits, but we could define recruits at the smolt life stage or really any life stage that we want. So hopefully that helps define that a little bit better.
Thank you. All right, take it away, Maureen.
Okay, great. So Ian brought up some really good points that I think it'll be demonstrated as I go through my presentation, and really how difficult it is to quantify the effects of hatchery fish on wild fish, particularly at the population scale. I think it's a lot easier at times to point to where there are potential for impacts, but it's really difficult to quantify what those impacts are. And when I was working with the Fish and Wildlife Service on this project, I would often get asked that question about, are hatchery fish bad? Are the hatchery fish in Eagle Creek, are they bad versus the wild fish? And it's really not a black and white issue. There's a lot of gray. These systems are dynamic and constantly changing. So yeah, it can be difficult to really quantify and demonstrate those effects at the population scale. But I'll get into that in a minute. I just want to say good afternoon. Today, I'm going to talk about some of the hatchery assessment studies that I conducted when I worked for the U.S. Fish and Wildlife Service and worked with the Eagle Creek National Fish Hatchery. And I have this little disclaimer at the bottom of 1 or 2 of my slides. And as David mentioned when he introduced me, I no longer work for the Fish and Wildlife Service. I work for Bonneville Power Administration. And a lot of the folks that I worked with on these projects have, like myself, they've moved on to different organizations or they've retired. So, I just want to say that the information and views that I'm presenting today, all the data and the results that I'll be presenting have been publicly or are in peer-reviewed and or annual reports. So there's nothing in here that's new or different, but that I'm not representing the Fish and Wildlife Service or any other federal or state agency today. So just really quickly, this is maybe more of a guide than an agenda, but it's just some areas that I'm going to touch on. I'm going to introduce myself a little bit in the next slide, and then talk about the kind of the history of hatchery production programs at Eagle Creek National Fish Hatchery, a little bit about the ESA listings of fish in Eagle Creek, and a timeline for when Fish and Wildlife Service got involved in monitoring and evaluating the hatchery and wild fish populations, and how that rolled into a 5-year comprehensive study that we call the Eagle Creek Ecological and Genetic Interactions Project. And then touching on some other work that we did looking at comparative survival, abundance, and growth of juvenile steelhead in Eagle and North Fork Eagle Creeks. And then just a recap or a summary, and I've got a slide with some links to literature and resources. So a little bit about me. David did a great introduction at the beginning. I do have a BA from Warren Wilson College in Asheville, North Carolina, an MS in biology from Western Carolina University. Following my graduation, I worked at actually a state trout hatchery in North Carolina. And then I went on and worked for the National Park Service in Smoky Mountains National Park, as well as Yellowstone National Park, doing inventory and monitoring of fish populations in park waters. In late 2004, I moved to Portland and started working for the U.S. Fish and Wildlife Service out of what's now known as the Columbia River Fish and Wildlife Conservation Office. And I worked there for 10, 11 years, conducting studies on hatchery wildfish interactions. And then in 2015, I moved from Fish and Wildlife Service over to Bonneville Power Administration, where I've been working as a hatchery policy lead for Bonneville's hatchery program. Garth, as I mentioned, Garth contacted me a couple of months ago and asked if I would be willing to give this presentation. So it's really just a fun jog down memory lane. I haven't thought about this project or a lot of this data in a while. So I'm happy to be here today. So I'm guessing that most of you on the phone know that Eagle Creek is located in the Clackamas River watershed. The hatchery is located about 16 kilometers upstream from the mouth of the Clackamas River. The hatchery was constructed in 1956 with the primary role of supporting commercial and recreational fisheries in the basin, and that's authorized under the Mitchell Act. And the hatchery is funded funding for the hatchery program is administered by NOAA Fisheries. So early operations and production programs at the facility included Thule fall Chinook salmon, and that production program was terminated in 1968. The hatchery also had a spring Chinook program that was terminated in 1987, and currently it's operating coho salmon and winter steelhead. So more recently, the hatchery has propagated winter run steelhead and spring Chinook under an agreement with Oregon Department of Fish and Wildlife. And the hatchery also annually rears and releases yearling coho salmon into Eagle Creek, as well as provides coho salmon to the Yakima Nation and Nez Perce tribes to support reintroduction efforts above Bonneville Dam. Now, as I was kind of preparing and getting ready for this presentation, I went through the U.S. Fish and Wildlife Service's publications in the Columbia River office and found the annual report, the 2021 annual report for the hatchery programs. And I came to find out that the memorandum of agreement, or the agreement that Fish and Wildlife Service had with ODF&W for fish production in Eagle Creek expired in May of 2021. And Fish and Wildlife Service did not renew that agreement. So essentially, it's my understanding that the winter steelhead and spring Chinook programs at Eagle Creek are terminated for now. So Eagle Creek, like many other hatcheries in the Columbia River Basin, especially before ESA listings of salmon and steelhead, used an out-of-basin broodstock for their hatchery program. Production at the hatchery was initiated in the early days, actually, by spawning native late-run Eagle Creek stock until about 1965. And then the program switched to this out-of-basin stock, and it's currently this Big Creek stock out of Washington. Now, I think when ODF&W took over the program, they reintegrated using a native stock for their brood stock. But when I was evaluating work within the basin, the hatchery was producing fish from using that out of basin brood stock, or what's called like a segregated program. So in the 1990s, many populations of salmon and steelhead, including the Eagle Eagle Creek late run steelhead, as well as the lower Columbia River coho, were listed as threatened under the ESA. And following this era of salmon listings, there was really a paradigm shift in hatchery programs and a transition from a fish production focus to an ecosystem management focus. So the emphasis was on conserving wild and naturally spawning hatchery stocks. And so for a lot of artificial production programs in hatcheries, they were taking on a new role to assist in species recovery. So as a result of the listing and then biological opinions with main stem Columbia River dams, the Fish and Wildlife Service allocated money to look at ecological interactions between hatchery and wild salmon and steelhead populations for several of the fish hatchery programs that the Fish and Wildlife Service operates. The Eagle Creek project was initially started in 2003, and that was with funding to look at juvenile migration timing from the hatchery to the Clackamas River. And then in 2004, Fish and Wildlife Service allocated funding for a more comprehensive evaluation of the program's impacts to wild fish. So I mentioned the paradigm shift in how artificial production is viewed after an ESA listing. And then there's this broader understanding that hatchery supplementation programs can have negative genetic and ecological effects on native stocks. And when you have hatchery programs that are operating in watersheds where ESA listed fish are present, there's an additional concern. So when fish are released from the hatchery, these stock salmonids can potentially interact with wild fish, competing for resources. There's the potential for genetic integration, and there's also concerns about how the hatchery fish will affect overall growth, survival, and fitness of the wild population. So to effectively manage ESA listed populations and watersheds where hatchery fish are present, it really requires that we collect some basic information on population abundance, life history, and genetic structure of the listed population. So at the time that we initiated the Eagle Creek studies, there was limited information on the population structure and really limited information on genetic contribution of hatchery and wild fish in Eagle Creek. So in these Eagle Creek studies, we systematically collected information on wild and hatchery steelhead populations, and then to a lesser extent on coho, to help adaptively manage the hatchery program and to reduce the likelihood of impacting wild fish. So the objectives of the studies that I'm going to talk about in the next few slides included collecting information on genetics, looking at population structure, including distribution and run timing of adults, as well as outmigration timing of juveniles, looking at what percentage of the steelhead population presents as residuals, or who essentially don't leave the fresh water and go into salt water. And then is there competition for habitat and differences in behavior between hatchery and wild fish in Eagle Creek? So I put this slide in the presentation to give you a general timeline for the studies that were conducted at Eagle Creek, and then to familiarize you with those who were involved in these efforts. The Fish and Wildlife Service formed a hatchery assessment team, which was a cross-organizational team and included Fish and Wildlife Service fish health staff, genetic staff, the hatchery support staff, and as well as biologists like myself who were in charge of implementing the monitoring and evaluation of the programs. So I mentioned 2003 is when we initially, or the Fish and Wildlife Service initially looked at the outmigration timing of juveniles. And then in 2004 to 2006, there was a rearing density study that the hatchery implemented. And that was to evaluate smolt to adult recoveries or smolt to adult returns for hatchery winter steelhead that were reared in low, medium, and high densities in the hatchery raceways. I didn't include any slides on that study in this presentation, but I'm happy to touch on that at the end. So from 2004 to 2009 is when the 5-year comprehensive evaluation of ecological and genetic interactions between hatchery and wild fish occurred. When we wrapped up with that study from 2010 to 2012, we learned a lot of information from the 2009 study, and that really was a springboard for asking some additional questions about these populations and comparing growth, survival, and abundance of steelhead in Eagle Creek and then in North Fork Eagle Creek. So this map shows a detailed image of the Eagle Creek study sites. As I mentioned, the hatchery is about 16 kilometers upstream from the confluence of Eagle Creek and the Clackamas River. There are 3 sets of falls on the river. There's the lower middle falls, which are downstream of the hatchery, and they have fish ladders that are maintained by the hatchery at both locations. The upper falls are located about a kilometer upstream from the hatchery, and those are impassable to migrating fish. So for the purposes of our study, we divided Eagle Creek into 3 main sampling reaches. The lower reach from the mouth to the lower ladder, the upper from the middle ladder to the hatchery, and then North Fork Eagle Creek was the 3rd reach. And North Fork Eagle Creek is, again, many of y'all are probably aware, is one of the main, or considered one of the main spawning tributaries to native Clackamas River late run winter steelhead. So this is just a real quick overview. We used a number of different methods to collect data in our studies. For several years, we implanted both juvenile and adult fish with radio tags. And these tags emit unique frequencies, and we use special antennas to track their movements and then collect information on where the adult and juvenile fish are distributing in the stream. We can gather out-migration timing for juveniles. So it's essentially how long it takes the juveniles to get from the hatchery to the mouth of Eagle Creek. We also conducted snorkel surveys and we tried using smolt traps to get estimates of fish abundance in the creek. We collected biological data and genetic samples from returning adults at the fish ladders. And then later in the study, we started using pit tag technology. And pit tags are essentially what's referred to as passive integrated transponders. And they're another type of tag that's used to track fish movement. They tend to be less expensive than the radio tags. And they also passively track the fish throughout the entire life, as long as you have a way or an antenna or a system to detect that pit tag. So I think, you know, one thing that when I was going back through some old slides from previous presentations, but it reminded me, we tried a lot of different methods to collect data in the creek, and it was really a lot of trial and error when conducting these types of evaluations. So I'm jumping straight to the next few slides are a little data heavy, but this table describes the travel time for juvenile steelhead from the hatchery to the mouth of Eagle Creek near Bonnie Lure Park. So we recorded the number of fish we tagged each year. We recorded the number that were detected at the mouth of Eagle Creek. And if those fish were detected at the mouth, they were then considered a migrant. We also recorded information on how long it took them to get from the hatchery to the mouth, and then the mean length of the fish. And I think the key takeaways from this table are, for some years, and those are indicated in red under the number migrated, less than 50% of the juvenile smolts that we tagged and were released from Eagle Creek National Fish Hatchery were detected at the mouth. So, you know, this indicates that there could be a lot of residual or non-migrating hatchery fish in this population, but it can also be a result of fish that have shed their tags or that they died before they reached the mouth of the creek. Another possible scenario is that the batteries on these radio tags died before the fish reached the mouth, and they weren't able to be detected by the receiver. So it paints a picture that, hey, it sends up some red flags that there's potentially a significant residual population that could be impacting wild juvenile fish in the creek. But when you sort of scratch the surface, there's a lot of other factors that could be contributing to the numbers that we're seeing. And I'll mention real quickly, we did radio tag coho smolts in 2003 and 2004. They left the system very quickly, and residuals are really less of a problem with cohos than with steelhead. So moving from talking about juvenile fish, the graphs on this slide represent the biweekly catch of adult steelhead at the lower fish ladder. So from 2005 to 2007, we trapped and inserted radio tags into adult steelhead at the lower fish ladder in Eagle Creek. And it was to determine where the fish were distributing and then the run timing of the adults. So on these graphs, the y-axis represents the percent of fish that were caught, and then the x-axis is our trapping period. The dotted line is the hatchery fish, and the solid line is wild fish. And I'll just mention the hatchery steelhead returned to Eagle Creek between December and March. So they're considered an early run winter steelhead, and then the native late run winter steelhead return typically March through June. And then the key takeaway from this slide is, our intended distribution should look more like 2006 and 2007. So what we really want to see is that there's no overlap in return timing between the hatchery and the wild fish. We want we're hoping or wanted to see the wild fish come in early. They're collected at the hatchery, they're removed from the system, they're reducing the risk for them to actually spawn in the creek. And then the wild fish, we want to see them coming in later in the season. So when we looked at 2005, that was, I don't know, it was atypical compared to the 2006 and 2007 return, but it definitely, again, was a cause of concern that, the fish are returning at the same time. There's the potential for the two populations to mix and compete for spawning habitat, or for integration to occur where hatchery and wild fish are spawning together. And this is the last graph, I promise. This represents habitat use and selection by hatchery and wild steelhead in Eagle Creek. So my colleague at the time did his master's thesis work on habitat use and competition between hatchery and wild fish in Eagle Creek. So this data and graph comes from that work. And just real quickly to orient you with the graph, we've got fish density on the y-axis, and then the habitat units starting from the mouth of Eagle Creek to the hatchery on the x-axis. And the gray line represents where North Fork Eagle Creek enters, and then the red line is where the middle ladder is located. So the bubbles correlate to the count of fish in a particular habitat unit. The bigger the bubbles, the more fish in that particular unit. And then the colors represent a particular habitat type, whether it's a glide, riffle, or a run. So the key takeaways from this, as we travel upstream, the creek, we're getting smaller, we're getting actually higher counts. So the closer we are to the hatchery, the more densely populated the area is with fish. So we got the highest densities, highest counts, and majority of habitat units containing fish were located in the upper reaches of Eagle Creek near the hatchery. So in 2000, the Fish and Wildlife Service conducted a genetic evaluation of late run winter steelhead and broodstock that was used in hatchery production. This study was really initiated in response to the ESA listings of the Columbia River or of the steelhead populations, and then concerns regarding using out-of-basin broodstock in the same watershed as the listed population. So Fish and Wildlife Service geneticists examined 4 populations of winter steelhead, the Clackamas River native population, the Eagle Creek native late run population, and then the Eagle Creek broodstock and the Big Creek hatchery stock. And the results from that 2000 study indicated that the hatchery stocks were more similar, more genetically similar to the other, and the wild stocks were more genetically similar. So what this does not really, this somewhat oversimplifies, I think, the genetic results and what we actually see with the population. We do know that the hatchery did use some native stock when it was, initially began operations in the late 50s. We know that they attempted to use a native stock in 2000 for 1 or 2 years. So there is likely some late run hatchery influence in that native population. So throughout the 2005 to 2012 study, we continued to collect genetic samples from adults and juveniles, and those samples were analyzed and assigned to either hatchery or natural origin fish using the baselines from that 2000 study that was conducted. So the results indicated that there was really minimal genetic integration between the hatchery and wild fish populations, and that the majority of natural production from hatchery fish was occurring in Upper Eagle Creek. In addition to the genetics work and then the population structure studies that we were doing, our fish health lab also examined hatchery fish before they were released from the hatchery, and then they did periodic sampling of wild fish in the stream. And this was done to identify, if the hatchery program posed a risk to wild fish. And the results from those studies indicated that fish released from Eagle Creek National Fish Hatchery posed a low risk of spreading disease to the wild steelhead and coho in the creek, and that they were overall primarily in really good health. So we didn't see a lot of risk of disease transmission between the hatchery and wild populations. So, all of this information is essentially pieces to a puzzle, and I think that was something that Ian used earlier in his presentation. But what does this mean in terms of impacts to fish? So it's a lot to digest on this screen, but what we did conclude from that 2009 study is, there were potentials for high levels of residual steelhead to remain in Eagle Creek from the hatchery releases. We did see in some years that there were spatial and temporal overlaps in return timing and distribution of adult hatchery and wild steelhead. We did observe hatchery fish spawning in the main stem, and that was confirmed when we, through our genetic studies, when we saw that there was a lot of hatchery influence in the natural population in Upper Eagle Creek. For habitat, we saw the highest densities counts and habitat units containing fish, again, located in the upper reaches of Eagle Creek near the hatchery. We saw through our radiotelemetry work, we saw that there was minimal straying of hatchery adults into North Fork Eagle Creek. I just mentioned we didn't see a whole lot of risk from disease transmission from the hatchery population. And then there was really very low genetic integration between hatchery and wild fish. So for management implications, it's great to have all this information, but you know, how is the hatchery operators, how are the hatchery managers going to apply that to their programs to help minimize these risks? And in working with the Fish and Wildlife Service and then the Eagle Creek hatchery operators and managers, we did recommend, and this recommendation was implemented, reducing the production levels for steelhead from 150,000 to 100,000, and then to a 350,000 coho smolt release. And the hatchery program did implement that in 2008. We also recommended that the hatchery spawn fish no later than mid-March to retain that early spawn and run timing, and then to keep the hatchery fish ladder open until April to remove hatchery fish from Eagle Creek. And what that means is previous to 2008, the hatchery has their a fish ladder where, you know, their broodstock come into holding ponds within the hatchery, and they were shutting that ladder down in late March, sometimes early April. And in 2005, we saw, you know, that there was a lot of fish, hatchery fish, that were coming back to the system later than we anticipated. And the fact that the hatchery ladder was closed, that may have contributed to them spawning in the wild. So the hatchery did implement keeping the ladder open for longer. And then we also recommended that they continue periodic sampling and monitoring of fish health in Eagle Creek. And I know that, and this is standard Fish and Wildlife Service policy, that all fish are sampled and monitored for disease before they're released from the hatchery. I don't know if the wild fish monitoring is still occurring at Eagle Creek, but I know that the hatchery fish are still being monitored. So, you know, we wrapped up that 2009 study, and I mentioned, you know, it opened up the door to more questions we had about comparative differences in survival, growth, and abundance between hatchery and wild fish in Upper Eagle Creek and in North Fork Eagle Creek. And, you know, just North Fork Eagle Creek had very little hatchery influence in the population, so it could be essentially referred to as a reference population. And then Upper Eagle Creek had a very strong hatchery influence. So in July of 2010, we initiated a 3-year study looking at comparative differences of steelhead in those two streams. So throughout the next 3 years, 2010 through 2012, we captured and tagged fish in North Fork Eagle Creek and Upper Eagle Creek between the middle ladder and just above the hatchery. Those fish were tagged with the pit tags. And then we went back over multiple years and within the season to try and recapture those fish, which helped us generate abundance estimates. We collected information on within season and over season growth, and then also survival estimates. You know, part of our efforts to estimate survival between the two streams included setting up the pit tag antennas. So we tag the fish, but then we have to have an antenna or a system to be able to detect those tags. The pictures I've got here just show, you know, we constructed these antennas within our own warehouse, and we did that in-house, as opposed to you can go through some of the pit tag. The folks who sell the pit tags, you know, do construct antennas, or they can. It's very costly, but we did this in-house. We installed the antennas in the stream. The picture down the bottom left shows us installing it towards the mouth of Eagle Creek. This was about 100 foot. I think these were 5 antennas that were about 20 feet in length. And so they were spanning the almost the entire bank to bank width of the stream. As most of you know, Eagle Creek's a really flashy system. So that picture to at least my right is showing that after one of a major storm or flood event, There was a lot of debris that came down through the system, and the antennas got destroyed. So we tried a few other times to build new antennas and reinstall them, and then to try and identify different locations for putting these antennas in. But our efforts were really were less than successful. So it was very challenging for us to be able to compare survival between the two streams, because we did have equipment failures. But in summary, kind of the results from that study indicated that estimates of population abundance in Upper Eagle Creek and in North Fork Eagle Creek varied considerably between sample years and study sites. Relative abundance of juvenile winter steelhead was greater in Upper Eagle Creek, and we saw that within the studies that we did between 2004 and 2009 as well. We didn't see any difference in over summer survival of juvenile steelhead between the two, between North Fork and Upper Eagle Creek. Average growth per day was significantly greater for steelhead in the upper section of Eagle Creek compared to North Fork Eagle Creek. But I think the main takeaway, or a main takeaway, is we did not find that freshwater growth, migration behavior, or survival in Upper Eagle Creek was negatively impacted by naturally spawning hatchery steelhead when we compared that to growth, survival, and behavior in North Fork Eagle Creek. And one other thing, I mentioned earlier the genetic studies from 2000 that indicated, that the native population was more similar to, or less similar to the hatchery population. Therefore, it could indicate that there isn't much integration happening between the two. Our geneticists went back, different geneticists went back later, looked at some additional studies, and found that evidence that the genetic baseline appears to contain evidence for hatchery integration. So, North Fork Eagle Creek, although it definitely has less of a hatchery influence than Eagle Creek, it's not necessarily this pristine production area for wild winter steelhead that we had been sort of characterizing it as. So I think that's just an important thing to note. And also that it's important to interpret that the genetic impact of the hatchery really needs to be looked at in context of the time frame for when the data is collected. So you're really telling a story. It's not necessarily just black or white. So, to summarize, there's been extensive research on hatcheries and interactions with native fish species in the Columbia River Basin. Eagle Creek is one of those studies, and we looked at it, over a 10, 11-year period and collected a lot of information that was useful, and it continues, I think, to help managers manage that program. And then just on a more global level, I think fish managers have updated hatchery strategies and operations throughout the Columbia River Basin to help improve culture, fish health, survival, fitness, and strain, with the objective of trying to meet the original purposes of providing sport commercial fisheries, but also helping to mitigate and reduce any impacts to wild fish. And finally, I think hatchery programs should be evaluated periodically to ensure that best management practices are observed and to reduce impacts to listed fish. And then the last slide is just some links to additional resources and literature on hatchery wild fish interactions. I've listed a link to the Columbia River Fish and Wildlife Conservation website, the Fish and Wildlife Service Pacific Region Hatchery Review website, and then as well as the Northwest Power Council's interactive hatchery map for hatcheries in the Columbia River Basin. Thank you.
Thanks, Maureen. That was excellent. Great overview, and Ian as well. Are there any questions for Maureen or for Ian and Maureen as you kind of assembled both of their presentations?
Does anybody have questions that you can feel free to unmute yourself or type it in the chat? I have one quick question probably for Maureen. You mentioned early in your presentation that the memorandum of agreement with ODFW expired, and I was wondering what is the implication of that?
Yeah, I don't, other than the fact that there won't be, the hatchery won't be rearing and releasing winter steelhead or spring Chinook salmon into Eagle Creek. I don't really know. I mean, that was news to me as well. And maybe Garth, you know, because he's working or actively working in the basin, has some more information on that. But I think one of the reasons that that Fish and Wildlife Service did not renew the agreement was the hatchery has a lot of deferred maintenance, some critical infrastructure projects that are that they need to actively work on. And so this could be just a temporary pause on those production programs, so the hatchery can you know, get the infrastructure updated, and then they'll resume those programs, but I just really don't know.
Hey, Gail, this is Garth. As I understand it, ODFW is still going to be putting hatchery winter steelhead and spring Chinook into Eagle Creek. They will acclimate those fish at Eagle Fern Park. They have a series of acclimation ponds there. So the production is not lost. It's just that Eagle Creek National will not be rearing those fish or managing the program. So it'll be entirely operated by ODFW at this point.
Okay, thank you.
I'd just like to say thank you to both Ian and Maureen for presenting with minimal arm twisting. Kind of had to reach back in the archives. Maureen, I know we haven't talked for a long time, and I really appreciate you stepping up to the plate and providing this presentation to folks for the Clackamas Basin. I think you and Ian both had a tremendous amount of value here for our constituents to get a better idea of hatchery wild interactions and just our fisheries that are in our backyard. So I sure appreciate it.
I just have one question, and it's kind of an overarching one, and that is, and I think a lot of folks have thought about this, is that for these organizations and whatnot that are concerned about this interaction between wild and hatchery fish and some of the proclamations that they've made, are they just overextending themselves on their conclusions, or what is it that, you know, they seem to get some traction by such, you know? It would just be interesting to hear your take on that, you know, because it's important, I think, for people to understand, you know, what we're coming away with here is that while there's more left to be learned, the jury is not out, it seems like, completely, you know, one way or the other. But yet some folks seem to argue that it is out. So I'd like to hear the takes on that.
Yeah, I'll just jump in real quick and just say, you know, I don't think it's a one size fits all. I think each, you know, I mentioned earlier that, you know, everyone or a lot of people want to, you know, are hatchery fish bad? They want this, you know, this really sort of black or white. Are they bad? Are they good? And it's just not that, it's not that simple. I think each watershed is unique, and whether or not a hatchery fish or a hatchery program is having a negative impact on wild or native stocks within that particular watershed is, it needs to be looked at, it needs to be evaluated, but you just can't paint a broad brushstroke over all hatchery fish and all hatchery programs are bad. You know, Ian mentioned earlier, they serve a purpose, not only to provide sport and commercial fishing opportunities. They're really important to our tribal partners and tribal cultural programs. So it's just, yeah, it's a tough one.
Yeah, and I think I would add to that just simply that I think it's important for those that are interested in this as a science issue, not to get caught up in the advocacy side of things. I think the groups out there that are advocating for particular policy prescriptions, I don't fault them at all for doing what they're doing. That's their stated mission. And so it's just important, I think, for folks that are interested in this topic from a science perspective to be able to learn enough about the issue to differentiate between what's sort of objective science perspective and what's just an entity or a person sort of advocating for a particular policy outcome.
Good, excellent. Any other questions?
Don't see any others.
All right, great. Well, again, thanks, Maureen and Ian. You did an awesome job. It's a topic that's certainly needed to be discussed, particularly in our basin, and you did an awesome job. So thank you for that. And with that, I want to just mention before folks go, just that we have our next session on January 11th, so 2 weeks from today, and I'll be back at our regular 6 p.m. time slot. So we'll continue our series focusing on water and fish, and we'll cover what fish field surveys are telling us about the fish populations and reproductive successes within our basins. So many of you know that's usually an ODFW mission that they do. And it's very important to understand what the actual observations are telling us, which Maureen alluded to in her presentation. So you can use the same Zoom link. And if you're already registered for this event, which I'm sure you were, you'll get further reminders. And again, thank you for attending tonight. And our thanks again to Ian and Maureen for today's wonderful talk. And we also thank our gold sponsors, Oregon Beverage Recycling Cooperative and the Clackamas Water Environment Services, and our bronze sponsors, the Clackamas River Water Providers, Metro, Port Blakely, and the Geological Society of the Oregon Country, helping us keep this conference free for everyone. But again, please consider donating if you want to donate to a great cause. Several more days. And thank you for joining us tonight. Please let us know what you thought of this afternoon. And we'll see you two Tuesdays from now, January 11th, and may you have a happy holiday. So thanks a lot.